Hello and welcome to the mitosis video lesson. In this video, we are going to go through a series of processes to finish the mitosis process. Interphase starts it all off by growing the cell to the point where it is able to grow through mitosis and split. In this picture, you see a cell as it is growing. Now we need to stop. We need to ask the cyclin if we can move on to the mitosis process. The cyclins are the overseers of all the jobs and processes happening inside of the cell. If the cyclin sees something not right, it will tell you to either go back and redo it, or it will just cancel out the whole thing. Now, it may not actually talk to you, but it tells your cells what they can and cannot do at the time. Now we're going to start the mitosis process because the cyclin said we were allowed to move on. As you can see in this picture, the first part of mitosis is called prophase. This is where the cell's DNA is diffused or dissolved, and the DNA is shown to the rest of the cell. If you look, this dotted line in a circle is typically your nucleus, where as now it is dissolving. We need to stop again and ask the cyclins if we can move on again. You have to do this for every single phase, going from interphase into the mitosis process of starting off prophase. You cannot start prophase until the cyclins allow you to. Now remember, the cyclins oversee everything to make sure that it does not mess up and that everything runs properly. After the cyclin allows us to move on from prophase, we decide to move into metaphase. This is the second phase of mitosis. This is where the sister chromatids, which are these X's in the middle, which was originally part of your DNA, they line up in the middle of the cell. It isn't very fun or interesting process, but they just move to the middle of the cell. They have to line up evenly to make sure that everything runs properly. Now we get to ask the cyclins if the metaphase process was done properly. If one of the sister chromatids does not meet up in the middle properly, the whole thing is messed up, because one side of the cell will get more than the other, which will not be fair, which will also mean that one cell will run too much and one cell will not run enough. After the sister chromatids line up in metaphase, the spindle fibers, which are these little things on the side, pull the sister chromatids apart from the centromeres. The centromeres is what holds the sister chromatids together. They are pulled to two opposite poles and ends of the cell. This process is what we call anaphase. This is also where they ask the cyclin to move on to the end of the mitosis process. This is pretty much one of the last times that we have to ask the cyclin to move on to the end of the mitosis process. The cyclins have done a very good job making sure that we have not messed up in this process so far. The last process of mitosis is known as telophase. This is the last process of mitosis, but it's not the last process of the cell. The phase is when a new cell wall forms in the middle of the two forming cells and separates them. When the sister chromatids got pulled apart in anaphase by these spindle fibers, a new cell wall starts forming in the middle. The sister chromatids will rejoin together with other sister chromatids and will start forming a new DNA. It will also form a new nucleus around. So we have officially finished mitosis, but we have not finished the entire cell cycle. After telophase separates the two and forms the new cell wall in the middle, the cell wall pulls the two cells apart. This is where the two cells form separately. This process is known as cytokinesis. Good job! We have officially completed the cell cycle with mitosis. Want to watch more? Watch the next video. The End by Nicholas J. Pruno. Photos complimentary of Google.